Once upon a time, in the land of Immunotopia, peace and harmony prevailed, orchestrated by the diligent council, the conventional dendritic cells. cDCs and their ability to initiate immune responses stood as the kingdom's defense against the insidious force of disease. But suddenly, Tumoria, a rebellious nation of invasive cancer cells, launched attack on Immunotopia, aiming to overthrow the balance maintained by the cDC nobility. These invaders are driven by a relentless desire to proliferate and dominate. The cDC-1 warriors wielded the legendary artifact known as the Gene Codex. This powerful tome contained the secrets of adaptive immunity and was important for cell-mediated responses that are the cornerstone of Immunotopia's defense. In the realm of cancer immunology, these dendritic cells are not just metaphorical characters, but pivotal orchestrators of the immune response against tumors. Their specialized roles in initiating and regulating immune reactions, particularly in the context of anti-tumor immunity, is important. We will now go over cDC development and function, cDC subtypes and expressions of BAF3, cancer hallmarks, anti-tumor therapies, and future directions.
In Immunotopia, there is a BAF3 badge that is only bestowed to a special kind of dendritic cell called cDC1s, who get the badge after the BAF3 training regimen during their development. The kingdom sages discovered enhancer elements at the IRF8 gene locus, which is critical for the noble cDC1 development. These enhancer elements are located 32 kb away from the transcription start site, and they increase the levels of IRF8 gene expression. BAF3 and IRF8 form a complex at the enchanted IRF8 32 kb enhancer site, binding to AICE motifs. This alliance is crucial for the development of cDC1s, propelling their growth and strengthening their abilities to combat threats to their kingdom. Beyond IRF8 stabilization, the BAF3 badge wields the power to influence other vital genes that provide the noble cDC1 with the strength to protect Immunotopia from any dangers.
The heroic dendritic cells are antigen-presenting cells that awaken T cells to defend the kingdom against the Tumorians. MHC class 1 molecules are like mirrors to showcase the inner beauty of the kingdom, presenting internal peptides rather than extracellular ones. Yet among the dendritic cells, a unique group named cDC1 possess extraordinary powers. It can showcase extracellular antigens on MHC class 1 mirrors, activating the CD8 T cells through cross-presentation magic. Cross-presentation occurs through two enchanting pathways. In the systolic pathway, extracellular antigens are transported through magical gates, such as receptor-mediated endocytosis, CLEC9A receptor, or engulfed through phagocytosis. The captured antigens are sent to the dungeon called endosomal vesicles, where they then cross into the cytosol through portals like ERAD. There, a grand proteasome processes these antigens into peptides, which are then placed onto MHC class 1 mirrors in the endoplasmic reticulum or the same phagosome. A transporter named TAP plays a key role in this process. On the other side of Immunotopia, in the vacuolar pathway, extracellular antigens are sent to the endosome dungeon, where proteases like the cathepsin S destroy the evil antigens into peptides, which are directly loaded onto MHC class 1 mirrors to be presented to CD8 T cells. Through this process, the dendritic cells ensure the safety of the kingdom from Tumoria by prepping the kingdom's defenders, the T cells.
In the grand strategy of the Immunotopia, the nuclear dendritic cells have diverse responsibility and capacities. Among them, the IRF8 dendritic cells were a regal lineage that include two distinguished branches, classical dendritic cells and plasmacytosis dendritic cells. These IRF8 dendritic cells were the guardians of tradition, upholding the ancient rights of cross-parentation and the summoning of the kingdom's defenders against the villainous cancer. On the other side, another fraction of squares exist, known as the IRF4 dendritic cells. These squares were renowned of their diversity, with distinct surface markers serving as symbols of their varied nature. Despite their varied appearance, the IR4 dendritic cells were united by their mastery over at least two powerful transcriptional programs. This program details strategy for producing various forms of immunological response that were targeted to combat the kingdom's numerous threats. For the specific group of IRF8 dendritic cells, they have the unique capacity to cross-present the valence Ds. By capturing fragments of the valence and presenting them to the assemble of warriors, they could reveal cancer's true form to the realm's protectors. BAF3 magic badge gives IRF8 dendritic cells core power. The magic badge can give an enchantment that enables users the ability to cross-present. The squads who wear this badge are from the BAF3 dependent squad. The IRF8 dendritic cell must get the help of magic badge of BAF3. Because without the badge of BAF3, the IRF8 dendritic cells lose their ability to perform the vital rite of cross-presentation. They will not be able to defeat Tumorians.
Immunotopia is in crisis. The rebellious Tumorian forces have evolved a strategy to invade the kingdom. The rebel forces are disguising themselves as normal cellular citizens of Immunotopia by wearing a concealing cloak filled with immune-inhibiting checkpoint proteins, like CTLA4 and PD-L1. By overexpressing these checkpoint proteins, the Temerians cast a bewitching spell on the T-cell warriors, rendering them powerless and incapable of recognizing the cancer cells, which can then proliferate and spread throughout the territory. An immune checkpoint is a ligand-receptor pair which can activate or inhibit an immune response. When checkpoint receptors on the surface of an immune cell recognize and bind to specific proteins or ligands on other cells, this can result in an inhibitory or stimulatory effect on immune responses. While this is a normal regulatory or tolerance mechanism, immune checkpoints can be taken advantage of by cancer cells to escape the immune system. This is a strategy the Temerian rebels have used, by overexpressing immune inhibitory proteins to prevent the activation of immune forces. So how will Immunotopia guard itself against the new threat they face from the Temerians expressing these immune deactivating proteins? Well, the capable mages of the land have devised a new magical artifact called immune checkpoint inhibitors. The noble cDCs can equip this artifact to prevent the T-cell soldiers from being tricked and deactivated by the Temerian rebels. There are two common artifacts, the Spear of Ipilimumab and the Divine Book of Nivolumab. These artifacts are anti-tumor therapies that block immune inhibitory proteins such as CTLA4 and PD-L1, stimulating an immune response against the cancer cells. CTLA4 receptor expression on T cells regulates T cell proliferation by competing with the CD28 receptor for binding to the B7 co-stimulatory ligand, which is usually present on antigen-presenting cells like cDCs. While CD28 stimulates B7 activity, CTLA4 inhibits it, thus preventing T-cell activation and proliferation. Cancer cells exploit this mechanism by overexpressing CTLA4 to evade immune responses. Immune checkpoint inhibitors, or ICIs, like ipilimumab, inhibit CTLA4, allowing B7 to activate T-cells effectively. Similarly, the PD-1 receptor binds to PD-L1, inhibiting T-cell activation. ICIs such as nivolumab block PD-L1 activity, preventing T-cell inhibition. These drugs counteract inhibitory pathways exploited by cancer cells, thereby enhancing immune responses and thwarting immune evasion. Through the power of the ipilimumab and nivolumab artifacts, the noble cDC cells successfully uncover the Temerian infiltrators, freeing the T-cell soldiers from the bewitching spell. The T-cell soldiers could now be activated and attack and destroy the Temerian cell forces, re-establishing peace across Immunotopia.
There is a subdivision of the dendritic cell nobility which depend on the BAT-F3 training to promote their anti-tumor abilities. With the help of another member of the royal family, CD103 positive dendritic cells, which undergo special BAT-F3 training, the army of the BAT-F3 dependent dendritic cell nobility can fight the Tumorian rebellion. The simultaneous action from BAT-F3 and CD103 positive dendritic cells fight off the Tumorians by causing tumor regression, allowing for the infiltration of T-cell soldiers into the Tumorian army to capture evil antigens, and cross-priming tumor-specific CD8 positive T-cell soldiers. This initiates Immunotopia's response against the Tumorian invasion. The noble dendritic cell then takes up evil antigens from tumor debris, mediating cytotoxic T lymphocyte priming at cancerous tissue. This amplifies the targets for immunostimulatory monoclonal antibody potions and increases anti-tumor priming abilities. When amplified, these MABs become stronger at rejecting the invasion. The Schools of the Kingdom by Sanchez Paulette et al. States that the noble BAT-F3 dependent dendritic cells are key for starting the Immunotopia response by immunostimulatory MABs.
Another tool the Kingdom of Immunotopia holds is their super secret cancer vaccine elixir. This magical potion helps prime T-cell soldiers for battle before they ever encounter the real enemy. It activates the soldiers and specifically prepares them to fight cancer troops. The elixir is made of three vital components: tumor lysate, GM-CSF, and TLR antigens, whose release is mediated by a PLG matrix. T-cells bind the tumor lysate or fallen cancer weapons and allow them to prepare to attack any other combatants they find holding those weapons. The T-cells, however, cannot prepare for battle alone. As discussed earlier, they require the assistance of noble dendritic cells to perform cross-presentation with the elixir and activate them for training. This requires a coordinated effort on part of the dendritic cells with the correct combination of cDC counselors. That's why the GM-CSF and TLRs are required. GM-CSF recruits dendritic cells to the vaccine training site, and TLR antigens act as messengers to the DCs, telling them to initiate cross-presentation. More specifically, stimulation via the TLRs upregulates DC co-expression of MHC class II and CD86, which increases their ability to train cancer-specific T-cell soldiers.